Hello everyone, this is Fusun Ozer from Ankara, Turkey. We will continue with ancient sheep again, and this time I will take you a different geographical location to Anatolia. Let me share my screen with you. Okay. Sheep was one of the four main animal species managed and domesticated in Southwest Asia during the Neolithic transition, when hunter-gatherer societies gradually switched to a sedentary life. And we know that sheep was domesticated within the area stretching from central Anatolia to Iran, shown with the blue shade on this domestication center map. You know, from the archaeological evidence such as corralling, which means keeping sheep in enclosures, and young male kill-off patterns, that sheep was already being managed by 9,000 to 8,000 BC from central Anatolia to Iran. After 7,500 BC, domesticated sheep becomes widespread, as evidenced from size changes and increased young male kill-off patterns in Anatolia, especially seen in very prominent Neolithic site, Çatalhöyük. After 7,000 BC, domesticated sheep was everywhere, and it was spread to Europe, Asia, and other regions, along with various elements of Neolithic. So in this slide, we see a depiction of Çatalhöyük. See, these are the corrals, enclosures where they keep the animals. So although sheep is the earliest farm animal domesticated, little is known about its demographic history. What we know so far is mostly come from modern sheep studies using mostly mitochondrial DNA and fairly recently from nuclear DNA. Let's look at what we know from mitochondrial DNA. There are 5 major haplogroups A to E in sheep. All 5 are seen in modern Turkish breeds. We know that haplogroup A is the most frequent in Asia, and haplogroup B is most dominant in Europe. When we look at Anatolia, we see a similar pattern. At the Western Anatolia, haplogroup B frequency is higher, whereas in the East Anatolia, haplogroup A frequency is higher compared to B. So high diversity that we observe in mitochondrial DNA would be consistent with scenarios where domestication involved multiple centers of domestication or a large and heterogeneous wild population in the single domestication center. So let's go look at the genome-wide polymorphism data. What does it tell us? So Kijaz and his colleagues studied 74 breeds from all over the world and genotyped 1,612 animals at 20,000 SNPs. And then when they plotted their data on a PCA, so here this PCA shows dichotomy of modern sheep breeds into Eastern clade, where Asian, African, as well as Eastern Mediterranean breeds cluster. And then in the Western clade, all the European from North Europe, Central Europe, and South Europe cluster together. So there is as we see, there is a clear distinction between Asian and European sheep, which may also imply multiple domestication or wild admixture events. At the same time, genomic analysis suggests high degrees of allele sharing across domestic sheep breeds. And this also has been considered evidence for the recent spread of sheep with desired traits across the globe, especially within the last 5 millennia as part of the secondary products revolution, such as spreading the wool sheep around Bronze Age. So we currently lack a solid demographic history model to explain these observations, such as high diversity, clear genetic structure, and recent coalescence times. What is missing is genetic data on the initial steps of domestication and characterization of the early domesticated sheep gene pool. In order to understand or help to understand sheep demographic history, we screened samples from Anatolia, basically from 6 sites, which are shown in red color here. So Pınarbaşı here is the oldest settlement. It's a rock shelter from where we got both Epipaleolithic and Neolithic samples. Hence, both wild sheep samples and domestic sheep or sheep in the process of domestication are available. So Boncuklu Höyük is an early Neolithic settlement exhibiting characteristics of a sedentary community. Tebecik Çiftlik, Can Hasan, Ulucak, and Barcın are later periods where there is abundant domestic sheep remains. So we obtained mandible and long bones, such as metapodia, from these sites. And 4 sites shown in black here are Central Anatolian and Southeast Anatolian sites where signs of herd management and domestication were clearly observed. So when once we got the samples to the clean lab, we cleaned the outer surfaces of the bones and cut a piece out. We UV them to decontaminate, and we grind them into a fine powder and then extract DNA from them. For the mitochondrial DNA study, we amplified a very short fragment, which is 144 base pairs long. This lies in the control region. Where we amplify a short fragment because this fragment is good enough to assign haplogroups. We obtained 3 replicates of PCR for each sample to validate the authenticity of the DNA. So once we get the PCR products, we sequence them in both forward and reverse directions. After editing them, we align them to reference Ovis area's mitogenome and assign the haplogroups based on these nucleotide positions. And each haplogroup shows a different combination of these nucleotides. So in total, we have studied 178 individuals. And our success rate, overall success rate, was 41%. Unfortunately, we cannot get DNA out of every single sample. Anatolia has a hot climate, which basically causes DNA to degrade. Heat and humidity is basically the enemy of DNA in these old samples. So once we assign the haplogroups, we examine the haplogroup diversity from Epipaleolithic to present day. And what we see is, we know that in the present day, we see all 5 haplogroups. So starting from Epipaleolithic, first we see the haplogroup B. These different shapes indicate different haplotypes, but I'm not going to put much emphasis on it because we're looking at a very short fragment. We have to be cautious when we're making inferences about it. So all I want to say about this slide is what we see is a gradual increase in the haplogroup diversity. And then as we come to the present day, we start seeing all 5 haplogroups. So basically, this implies that through time, the haplogroups are added from other domestication centers or through introgression from the wild. Okay, so when we compare these ancient haplogroup results with Europe and Asia, again, we see in Europe haplogroup B frequency is higher, and then in Asia, haplogroup A frequency is higher. So when we did goodness of fit test to see if haplogroup frequencies are different between Anatolian Europe and Anatolian Asia, what we saw is there is no significant difference between Europe and Anatolia, whereas the haplogroup frequency differences are significantly different between Anatolia and Asia. So which tells us that Anatolia shows similar patterns of haplogroup diversity to Europe. So this is all I want to say about the mitochondrial DNA part. But since mitochondrial DNA reflects only maternal history, we decided to take this study to further and investigate genome-wide variation. So we prepared whole genome libraries from the DNA we extracted. And we shotgun sequenced the libraries. By the way, we prepared libraries for only 36 individuals. We shotgun sequenced them, and once we get the results, we analyze them in the dry lab. So if our libraries had more than 1% sheep DNA, we continued. We either deep sequenced the same libraries or we captured SNPs from them. In our case, we did both. We deep sequenced and captured 20,000 SNPs from our libraries. Again, once we get the data, we analyzed it in the dry lab to understand the demographic history and like selection patterns and some other population genetics analysis. Okay, so out of 36 whole genome libraries, we could only get moderate quality data from 4 individuals. 3 of these came from this settlement called Tepecik Çiftliköyü, and 1 genome from this western side. So genome coverages ranged between 0.022 to 0.273. Our number of SNPs, again, were really modest. So when we get the data, we check for the authenticity. One of the ways to check authenticity is look at the post-mortem changes at the ends of the reads, at the ends of the fragments we obtain. We expect to see some post-mortem degradation in ancient DNA fragments. Those are basically in the form of C to T transitions. And if their percentage is somewhere between 20 to 30, this is acceptable level, and which means these reads are authentic. So these profiles belong to the sheep we studied, and I mean, they clearly show a signal of authenticity. And we use molecular data to assign the sexes. This is one of the important issues for archaeologists, especially zooarchaeologists, because sometimes it's hard to tell sex from the morphological data or using the bones. So here we used autosomes and X chromosomes. The red dot shows the X chromosome ratio. If it's close to this blue line, that means the individual is female. And if it's away from the blue line, out of the confidence interval, then it is a male, since males carry only 1 X chromosome. Okay. So once we get our genome data, what we did was we plotted them on a principal component. First, we used the modern data to construct the PCA, and then we projected our ancient genomes on it, as seen here. So what we see in this PCA is, these are our ancient individuals, and the reddish breeds are the European breeds, the cluster for the European breeds, and then the bluish breeds are the Asian ones. So our ancient individuals have a central position between Asia and Europe, but much closer to the European breeds. So we wanted to validate these patterns we see on PCA. So what we did was we conducted some D-tests. In the first one, we looked at if all ancient sheep are equally distant to each other, and our D-test results show that yes, they are equally distant to each other. No specific ancient individual picks a particular one, so they're all equally similar. And then we checked if any ancient sheep picks a modern breed. And out of 216 tests, 89% of them showed that Anatolian Neolithic sheep picks each other than moderns. So again, we tested if any modern breed picks a particular ancient sheep, but again, they were equally distant to each ANS. No modern breed picked one Anatolian Neolithic sheep at each instances. Okay, so we also tested if Anatolian Neolithic sheep prefers European or non-European breeds. 49% of the 80 tests were significant, and they showed that Anatolian Neolithic sheep had higher affinity to European breeds. When we looked at further into Europe, we showed that Anatolian Neolithic sheep has higher affinity to Central and Northern Europe than to South European breeds, which tells us that South European breeds might have differentiated later on due to migrations from Asia. And this was indeed shown in other studies where they say during the Bronze Age, wool sheep was, or sheep selected for its wool was migrated to Europe. And then our last D-test was, we performed 306 comparisons to see if modern breeds, a modern breed choose a modern breed or ancient Neolithic sheep. We saw that modern breeds consistently choose other modern breeds over Anatolian Neolithic sheep. So another test we ran was admixture. So here we see our ancient individuals. These are the European breeds and then the Asian breeds. When we look at the ancestry components, for example, pink here shows the Europe and then the blue, basically the Asian component. So as we go further, we see that like our ancient individuals show similar ancestral components with the Europe, especially with the Central and South Europe. Next, we conducted F3 statistics to see which European or Asian breeds do our Anatolian Neolithic sheep show genetic affinity to. The warmer the color, the higher the genetic affinity, and then the colder the color, it's less genetic affinity. So we see that our Anatolian Neolithic sheep shows higher affinity to European breeds, especially to Central European breeds, Northern and Central European breeds. So this is pretty consistent with what was seen in the literature for pig and goat before. So they report that For example, pigs were transferred or migrated to Europe through the Danubian route. And this probably is the case for our sheep, since they show higher affinity to Central and Northern Europe. So in conclusion, for the mitochondrial results, we see that there is a increase in the haplogroup diversity after 7000 BC, which is compatible with admixture from other domestication centers or from wild populations. We see that haplogroup frequencies are not statistically different from Europe, but they are different from Asia. So this tells us that Anatolian gene pool is similar to Europe. And then our genome-wide polymorphism data shows that Anatolian Neolithic sheep are genetically closest to present-day European breeds, and especially those from Central and North Europe. And since South Europe seems a bit differentiated, as I said before, Asian contribution to South European breeds, possibly during the Bronze Age, might explain this pattern we see. And finally, there are many people to thank for their contribution to this study. There are two ancient DNA teams in Ankara, one in Hacettepe University and one in Middle East Technical University, and we are two sister laboratories working together. And also, I would like to thank to the organization committee for their kind invitation to present our work here. Thank you.